Hi students, welcome to E-Pathashala program. I am Dr. K. Bhaskarachari, working as a senior scientist in the National Institute of Nutrition, Hyderabad. Today we are going to discuss about the gut bacteria, that is intestinal microflora, and its uses and how they could be useful for preventing certain diseases and also the pathogenic bacteria which creates problems in the human body. As we all knew that human intestine, you will be shocked to know that more than 2 kilograms of bacteria is existing in our body, in the gut or in the intestine. Most of the bacteria which is existing in the human gut is very useful and symbiotically it lives. Also, when there is a good, there is a bad also, you could have some pathogenic bacteria which create problems in the human body. Therefore, when pathogenic bacteria enters, doctors will treat with antibiotics. But when antibiotics were treated, the human body, there is an every possibility of the good bacteria also will get killed. So, therefore, supplying the good bacteria in the form of either prebiotics or probiotics or symbiotics are very important. But to know about the gut microbiome and its genome and its effects on the human body, we must learn what sort of bacteria which is propagating, which is living in our gut for so many millions of years when man started his travel from millions and trillion years ago. So, these bacteria will try to help, and some of the bacteria even synthesizes the micronutrients which are helpful to the human body. So, also they will also consume some of the food ingredients and they could convert them to the useful parts. For example, soybean has dioxin, genistein like molecules which are powerful antioxidants. Unless they become an active form, they cannot be useful in the human body. The active form can be done by the bacteria which is living in the human gut. That is why human gut bacteria has a beneficial role and they will do lot of useful things for the human body. So, what we will learn from this module? What are the objectives of this thing if you really listen to this lesson? You will understand the concept of gut microbiota and learn about the gut microbiota profile during infancy and adult life. Also, we understand the critical role of gut microbiota in a few important functions. We will also understand the role of gut microbiota in human disorders. Module on general functions of gut or intestinal microflora. The human gut hosts as many as over 1000 species of bacterial species which collectively code over 5 million genes. The phenomenal size of gut active microbiota is suggestive of its role in human health. With over 10 to the power of 14 microbes, the gut microflora accounts for as much as 2 kilograms of human weight. So, look at that, what we were thinking only bacteria is a small size, but if you put all the bacteria on a balance, it will weigh around 2 kilograms. Given the volumetric contributions, its collectively vast genome and their physiological importance, the gut microflora is often perceived as a functionally important organ. Gut microflora plays an important role in many aspects of human physiology such as immunity, nutrient metabolism, immune modulation and drug metabolism, in addition to involvement in diseases. Recent research indicates that alterations in microflora profile is associated with many disorders, and understanding physiological effect of gut microflora may pave the way for newer ways of treating several human disorders. The term microbiota describes the community of microbes living in a particular environment. Therefore, the gut microbiota can be defined as the community of microorganisms inhabiting the gut system. Human beings are born sterile. When we come out of the mother, we are sterile. But microbial colonization begins soon after the birth. The moment we come from the mother's womb, the bacterial inhabitation starts occurring in the infant body. Microbial profile of human being depends on age, dietary pattern, environment and medical treatment regimens, etc. In infants up to 3 years, the gut microbiota is mostly represented by actinobacteria, followed by proteobacteria, firmicutes and bacteroidetes. In adults, most of the gut microflora consists of firmicutes, bacteroidetes, followed by actinobacteria, proteobacteria. There is a difference between infancy and in the adults also. It has been now unequivocally established that interaction with gut microflora is essential for physiological balance, as they play an important role in multitude of body functions, and alterations in their profile is associated with many disorders. Present estimates suggest that the collective human gut microflora is composed of over 35,000 bacterial species. See the beauty. As a human being you are one, but how many species of bacteria are living in your gut is more than 35,000 bacterial species, and put together quantity wise they are around 2 kilograms in the adult human body. A Danish study of the gut microbiome and their function involving 123 non-obese and 169 obese individuals led to a concept of high gene count and low gene count. The high gene count and low gene count individuals are differently susceptible to diseases. So when they have worked in the Danish study, they have found this HGC and LGC. Anatrotroncus colihominis, Vitrio vibrio crossotex, Achaemenacea species and Fecalibacterium species are common representatives of the high gene count gut microbiota with a high Achaemenacea ruminococcus torque Gnavus ratio. High gene count individuals show higher levels of butyrate production and are less susceptible to metabolic syndrome and obesity. See, this is the beauty, that you could check the species of the bacteria and you can come to a conclusion what sort of a problem a person may occur in future also, which you could predict by looking at the bacterial species. Conversely, LGC, that is low gene count individuals, host a higher proportion of pro-inflammatory bacteria, that is bacteroids and ruminococcus gnavus, that are common cause of inflammatory bowel disease. Bacteria such as parabacteroids, Campylobacter, Dialister, Porphyromonas, Staphylococcus and Anerostypes are some of the common inhabitants found in low gene count individuals. The predominant phyla that inhabit the large intestine harbors more than 70% of all microbes are firmicutes and bacteroidetes, as well as primary pathogens such as Campylobacter jejuni, Salmonella enterica, Vibrio chlorella, Escherichia coli and Bacteroides fragilis. The gut microbiota also exhibits both temporal and spatial differences in distribution. Streptococcus is the predominant genus in the distal esophagus, duodenum and jejunum, while Helicobacter is the predominant genera of the stomach. You could see the difference between what sort of bacteria will be in the stomach, what sort of bacteria will be in the jejunum or duodenum. You should also understand the pH differences. If you take the difference between pH will be around 1 to 2 in the stomach, whereas in the duodenum and jejunum will be higher pH. So that the pH versus bacteria also one could really relate, understand its habitation. Helicobacter genera determines the entire microbial landscape of the gastric flora. When Helicobacter pylori inhabits the stomach commensal, there is a rich diversity constituted by other dominant genus such as Streptococcus, Prevertillo, Velionella and Rothia. This type of diversity is disturbed when Helicobacter gains pathogenicity. Then we learn about the functions of gut microflora. Gut microflora and host nutrition. Short chain fatty acids such as butyrate, propionate and acetate produced by bacterial fermentation of dietary fiber and undigested carbohydrate. Undigested carbohydrate means in addition to the dietary fiber, some of the starch also which is not digested, which will reach the large intestine, is called the resistant starch, which is also under the category of undigested carbohydrates, which also behaves like a dietary fiber in the distal part of the intestine, serve as a rich sources of energy for the host.
Many bacterial genes such as glycosyl transferases, glycoside hydrolases and polysaccharide lysases play an important role in bacterial handling of carbohydrates. The best example among these organisms is bacteroids Theta-Rheumatoin micron, which expresses more than 260 hydrolases. Microbes such as Axolobacter formigenes, Lactobacillus species and Bifidobacterium species reduce the risk of oxalate stones in kidney by utilizing oxalates in the intestinal milieu. You could also look at that when we talk about in the food and nutrition and the human nutrition, particularly when you talk about high oxalates food rich in like green leafy vegetables and people more prone for the kidney stones, we will generally tell the doctor will ask them to avoid high oxalate foods. So, but when oxalates are present in the foods and look at the symbiotic, this bacteria particularly, they are actually utilizing these oxalates so that they are trying to safeguard the kidney from not to going for a disease risk. Synthesis of vitamin K and several components of vitamin B complex is another major metabolic function of the gut microbiota. Members of the genus Bacteroids have been shown to synthesize conjugated linoleic acid that is known to be anti-diabetic, anti-atherogenic, anti-obesogenic, hypolipidemic and immune modulatory properties which conjugated linoleic acid has. Polyphenols. Polyphenols are the plant secondary metabolites. They are known as powerful antioxidants which are abundantly present in the foods, which usually remain inactive in the diet or biotransformed to active compounds after removal of the sugar moiety by the gut microbiota. See, aglycans will be formed from the glycosides. The glycosides is the sugar moiety which the bacteria will eat, and the aglycans will be released, or the bioactive forms of that particular polyphenols will be released. Recent studies have shown that human gut microbiota also help in breakdown of various polyphenols and thus facilitate bioavailability of compounds like flavonols, flavonones, flavontriols, anthocyanidins, isoflavones, flavones, lignans. Like such compounds, the bioavailability will be increased because of this bacteria. Gut bacteria also essential for the transformation of natural compounds to perform their bioactives. Example, lignans. Lignans are present in wide range of foods such as flaxseed, vegetable, fruit and beverages, even the sesame seeds also having these lignans. Lignans afford protection against cardiovascular diseases, hyperlipidemia, breast cancer, colon cancer, prostate cancer, osteoporosis and menopausal syndrome, dependent on the bioactivation of these compounds to enterolactone and enterodiol. Larsinosenolysis, one of the most abundant dietary lignan, and it can be demethylated and dehydroxylated by gut bacteria such as Peptostreptococcus, Seco-MT5M3 and Eger-Chalonce Seco-MT75M2.
Xenobiotic and drug metabolism. Capability of gut microbiome to metabolize xenobiotics and drugs was first recognized almost 40 years ago. An increasing body of evidence now provides sufficient insights on the role of gut microbiota on xenobiotic metabolism, which could have profound impact on therapy for various diseases in future. Gut microbial metabolites such as P-cresol may interfere with the ability of liver to metabolize certain xenobiotics such as acetaminophen due to as a result of sulfotransferase inhibition. Another interesting example of microbiome induced drug metabolism is the microbial beta glucuronidase induced deconjugation of the anti-cancer drug irinotecan that can be contribute to its toxicities such as diarrhea, inflammation and anorexia. So, if we look at the defense against pathogens or the host immunity, gut microbiota provides protection to host against colonization by pathogens that are associated with diseases. The mechanisms that regulate the ability of the microbiota to restrain pathogen growth are complex and include competitive metabolic interactions, localization to intestinal niches, and induction of host immune responses. Intestinal epithelial cell barrier is a major physical defense against pathogens, and gut microflora operate to strengthen and repair it. Another mechanism involved in the health benefits afforded by probiotics includes the formation of low molecular weight compounds such as organic acids and the production of antibacterial substances termed bacteriocins. Organic acids, in particular acetic acid and lactic acid,
are most commonly described antimicrobial compounds responsible for the inhibitory activity of probiotics against pathogens and are active against gram-negative bacteria. Bacteriocins produced by gram-positive bacteria. The common mechanism of bacteriocin-mediated killing include the destruction of target cells by pore formation and or inhibition of cell wall synthesis. For example, nisin forms a complex with the ultimate cell wall precursor, lipid 2, thereby inhibiting cell wall biosynthesis of mainly spore-forming bacilli. Antimicrobial produced by prebiotics. Example of lactolin, lactobacillin, lactobrevin. Like many of these prebiotics are produced as an antimicrobials. Commensal also prevent pathogen infection by altering host environmental conditions such as pH, which I have described you earlier about the pH and link with the bacteria. In the intestine, commensal bacteria generate short chain fatty acids which can alter the local pH to inhibit the growth of certain intestinal pathogens. See, that is the beauty, how the good bacteria are killing the bad bacteria by changing the pH. The short chain fatty acids butyrate is known to downregulate the expression of several virulence genes in Salmonella enteria, Seroverenteritis, Typhimurium. Fructose generated by fucosidase expressing commensal bacteria such as bacteroids, beta-micron, modulates the expression of the virulence factor LER, a master regulator of the locus of enterocytes effacement genes in EHEC28. Bifidobacterium blocks the colonization of pathogenic Escherichia coli by lowering the pH of the gut enteroecosystem. Commensal bacteria also prevent pathogen colonization and infection indirectly by enhancing the host defense mechanisms such as functionally promoting mucosal barrier function and enhancing either innate immune response. The promotion of epithelial barrier function by commensal bacteria is supported primarily by indirect evidence in which studies have demonstrated that germ-free mice and mice deficient in proteins involved in microbial recognition such as NOD2 and the TLR signaling adapter MYD88 have impaired the production of antimicrobial peptides, particularly by Paneth cells of the small intestine. Bacterial signaling likely through MYD88 also promotes barrier function through the production of secretory IgA that is released by intestinal epithelial cells and functions to bind the microbial antigens, neutralize pathogen activity and prevent infection. Gut bacteria, including bacteroides, fragilis and bacillus, are probably required for the normal development of GALT and mucosal immunity in all mammals. They are also required for somatic diversification of immunoglobulin genes. Gut microbacteria colonization induces a conspicuous response of the gut immune system to the production of IgA, which plays a critical role in regulation of gut bacterial communities in the small intestine. Another factor that may play an active role in the induction of local immune responses is the interleukins, which may function as sense bacteria. Studies of animals bred under germ-free conditions showed that germ-free animals presented morphological, structural and functional abnormalities including reduced vascularity, digestive enzyme activity, muscle wall thickness, cytokine production and serum immunoglobulin levels, smaller Peyer's patches and fewer intraepithelial lymphocytes. Microflora and diseases. Usually gut bacteria and the host live in a commensal manner. However, gut bacteria can be potentially harmful when the ecosystem undergoes abnormal changes characterized by relative increase in the population of pathogens. For example, antibiotic treatment and surgery cause pseudomembranous colitis due to toxin produced by Clostridium difficile and sepsis of Escherichia coli, Enterococcus faecalis and Enterococcus faecium, intra-abdominal abscesses due to Bacteroides fragilis. Imbalance in gut bacteria composition is associated with intestinal symptoms such as bloating, abdominal pain and diarrhea. The abundance of bifidobacteria may have a negative correlation with abdominal pain. Besides, many other diseases are likely to be related to gut bacteria such as inflammatory bowel diseases, obesity and diabetes, liver diseases, chronic heart diseases, cancers and even autism. Gut bacteria and inflammatory bowel disease. Inflammatory bowel diseases is a disease of common occurrence. Reciprocal interaction between commensal gut bacteria and the host may induce allergies, inflammatory bowel disease and disorders in bacterial recognition by macrophages are strongly related to the pathogenesis of IBD, that is inflammatory bowel disease. Ulcerative colitis is one of the two major idiopathic IBDs. In UC patients, the disease is limited to the colon. Number of lactobacilli drops significantly during this active phase of the disease, and denaturing gradient gel electrophoresis analysis suggests that Lactobacillus salivarius, Lactobacillus manihopivorians, and Pediococcus acidulolacticea were present in the remission but not during active inflammation. Bacteria of Clostridales group are the most prominent in samples from the inflamed colon, indicating this bacteria might accumulate during colitis. Bacterial products exacerbate acute inflammation via TLR2, TLR4 and signaling potentially trigger TLR dependent accumulation of neutrophils and T cells. Crohn's disease is another type of IBD. It has been thought to be an autoimmune disease in which the body immune system attacks by the gastrointestinal tract and causes inflammation. It has been found that the fecal microflora in patients with both inactive and active colonic CD contained significantly more enterobacteria than in healthy subjects. In addition, about 30% of the dominant bacteria did not belong to the usual dominant phylogenetic groups. Another study found that 5 bacterial species characterized by dysbiosis in CD patients were a decrease in dilastinovenses and an uncharacterized species of Clostridium cluster XIVA, a fecal bacterium processus and bifidobacterium, and an increase in Ruminococcus granulosa. There was a different composition of gut microbiota in unaffected relatives of patients with CD compared with healthy controls. In addition, an increased abundance in bacteria including enterobacteria, Pasteur venenos, like many other bacteria, which are correlated strongly with the disease status in a large pediatric CD cohort study. Gut bacteria and obesity link also with the gut bacteria. Normal gut bacteria play an important role in diet-induced obesity because germ-free mice have been reported to be thinner and did not become obese when subjected to high-fat diet. The high-fat diet altered the composition of bacteria to display higher levels of luminal firmicutes and proteobacteria and lower levels of bacteroidetes, indicating that obesity may be associated with decreased diversity and changes in composition of the gut bacteria. The ratio of firmicutes to bacteroids has been found to be correlated to body weight, with the ratio being higher in obese people. Gut bacteria could also affect obesity by promoting chronic inflammatory status. Gut bacteria may affect obesity through regulation of the microbiota brain-gut axis by its metabolites. Overweight individuals have more fecal short-chain fatty acids than lean individuals, with significantly increased levels of propionate. Butyrate and propionate were reported to protect against diet-induced obesity and regulate gut hormones. Some human bacteria have the ability to produce conjugated linolenic acid, which is known for anti-obesity outcomes. It is important to note that the amount of conjugated linolenic acid found in human adipose tissue is thought to be directly related to the dietary intake. Members of Bifidobacterium breve, Bifidobacterium bifidum, Bifidobacterium pseudolongum are known to produce conjugated linolenic acid. Lactobacillus rhamnosus PL60 is a human originated bacterium that produces T10C12 type of conjugated linoleic acid. A study showed that L. rhamnosus PL60 reduced the body weight in obese mice fed obesogenic diet without reducing energy intake and caused a significant reduction of white adipose tissue. Lactobacillus reuteri therapy prevented the pathology of abdominal obesity and age associated weight gain in mice. Gut bacteria and liver diseases. Gut bacteria play a key role in the maintenance of gut-liver axis health. Ethanol, ammonia and acetaldehyde produced by the intestinal microflora are generally metabolized by the liver and control Kupffer cells activity and cytokine production. Small intestinal bacteria overgrowth may be an important pathogenesis of non-alcoholic steatohepatitis. Small intestinal bacteria overgrowth in NASH rats decreases intestinal movement. Antibacterial treatment can alleviate the severity of NASH. In a study of Escherichia coli was excessively increased in the NASH rats, as was with the serum level of aminoferase. TNF-alpha may be an important mediator in the promotion of NASH by SIBO, which was supported by the fact that the level of aminoferase went up and down and the serum level of TNF-alpha. Recent evidence also indicates that gut microbiota is associated with alcoholic associated liver damage. Gut derived endotoxin and other luminal bacterial products may be cofactors for the development of alcoholic liver diseases. Gut bacteria and chronic heart diseases. Gut bacteria have a direct link with the risk of cardiovascular diseases. They form trimethylamine from dietary choline after its conversion to TMAN oxide in the liver. TMA could act as a pro-atherogenic compound. Intestinal concentration of mostly adherent bacteria in chronic heart failure patients increased and adherent invasive Escherichia coli were identified. Disturbed intestinal microcirculation and barrier function in chronic heart disease likely triggers cytokine generation, contributing to further impairment in cardiac function. Colonization with Lactobacillus brevis decreases bowel permeability, whereas colonization with Escherichia coli, Klebsiella pneumonia and Streptococcus viridien showed the opposite effect. It was suggested that bowel wall with high permeability may lead to bacteria and or endotoxin translocation, which may be an important stimulus for inflammatory cytokine to be activated in chronic heart failure. Hypercholesterolemia plays a key role in the development and progression of coronary heart disease. Dahl's salt-sensitive rats fed the commercially available probiotic product, Good Belly, which contains the leptin-suppressing bacteria, Lactobacillus plantarum 299V, resulted in decreasing circulating leptin levels, which is very important, leptin levels, smaller myocardial infarcts and greater recovery of post-ischemic mechanical function. See, this is the beauty of this experiment using the Dahl-sensitive salt rats fed the commercially available probiotic product. The effects were similar to the effects in rats treated with minimally absorbed antibiotic vancomycin. So, these results show that Lactobacillus plantarum strains may be beneficial for cardiovascular diseases. Thus, it is very important to understand the gut microbiome versus its health effects, and particularly when they are inhabited in the adulthood, and right from the childhood to the adulthood, the bacterial species are changing, and the effects of these things diet can be induced or by not only by probiotics but also with the prebiotics, which can improve the diet quality and by increasing the species as well as good bacteria in our gut for the human health.
Now, we will conclude the module which we have learned about the gut microbiota or the microflora, where the human body contains various microbiomes inside the gut, and these bacteria which are coexisting, and it is a beneficial effects. What we have learned so far has been against liver diseases, also against arthritis, and many of drug metabolizing enzymes, and also it will convert many of the compounds into bioactiveness. So, therefore, living with a good bacteria is beneficial, and therefore, we have to propagate this bacteria by various means, and we have to live healthily by living with the bacteria inside. But also we should remind that when a pathogenic bacteria enters into our body, which will create an havoc, therefore, such pathogenic bacteria should be removed using with a proper treatment by consulting the doctor. But what we have learned is this bacteria, which is also having its own genome, will also influence the human genome, and therefore, living with such a symbiotically living with a bacteria is very important to understand, and also we can design how to design the diet for improving the bacteria and the quality bacteria, particularly the number also very important for looking into various aspects of the metabolistic activities inside the body. Thank you.